Congratulations.
Thank you.
Kaiser has 75% teaching and 25% research appointment and is currently teaching 4 soil scientist classes from the 100 to 900 level, covering introduction into soil resources, urban soil properties, soil chemistry, and soil carbon and nitrogen dynamics. In his research activities, he is focused on clarification of effects of sustainable soil management practices on the amount, composition, and stabilization of organic matter, which is highly relevant for soil health, mitigation of climate change, and resource-efficient soil productivity. In Nebraska, his group established field experiments to analyze the synergistic effects of individual sustainable soil management strategies, such as the application of biochar and biosolids, cover cropping, and reduced tillage on soil carbon, nutrient, and water retention. Kaiser was born in I said it just right this moment, Dennigroden, a small town in central Germany. He earned a diploma in geoecology and a doctorate in natural sciences from the University of Potsdam, Germany. Prior to moving to Nebraska, Kaiser worked at the Leibniz Center for Agricultural Landscape Research near Berlin, Germany, and at the University of California, Merced, and the University of Kassel, Germany. But we are honored to have you here quite a bit. We've heard a lot about you, so it's finally, I'm happy to hear this. And I want to thank Sebastian Barrett, Dr. Sebastian Barrett, to the University of Minnesota for recommending Dr. Kaiser to give us a presentation here today. And with that, now without further ado, I'd like to introduce Michael Kaiser.
Thank you. So I'm sharing my screen. So let's see. Okay. Okay, here we go. So it starts at the end. I just flip through. Okay, can you see my slides?
Yes, we can see your slides.
Okay, then I'm starting. So again, so thanks for inviting me. It's a big honor and pleasure to present data of our research in the Char Talk today. Title is Application of Biochar to Improve Functioning of Agricultural Soil under Corn-Soybean and Insights from Field Experiments in Nebraska. Thanks for the introduction. So we had enough words about me. These are our partners supporting and funding our biochar work. So we have a hedge project. Nebraska Center for Energy and Science Research and Nebraska Public Power District are supporting us. City of Lincoln in Nebraska, Nebraska Forest Service, Nebraska Water Center, the USGS, and the Oregon Biochar Solution. They are all in some kind of support or funding or both involved in our projects. That is our team at UNL. Britt Fossum, that's a Ph.D. student. She's leading the field experiments in Nebraska. Dr. Katja Köhler-Kohl, soil health and extension management educator, and Dr. Arana Malaka, who is a research assistant professor and an expert in soil nitrogen and water dynamics and in nanotechnology. So that's our core team here at UNL. Some background and challenges. So why do we do this biochar work? First of all, over the last 300 years, increasing agricultural intensity. So we lost about 26% of organic carbon in the topsoil. It's up to 50% in the Great Plains. And that led to a large cumulative loss of soil organic carbon, which went as carbon dioxide into the atmosphere. And why does it matter? So soil organic carbon, soil organic matter improves soil aeration and water holding capacity, improves soil structure, aggregate stability, lowers bulk density and soil erosion. It's relevant for storage and supply of soil nutrients. It's substrate for soil microorganisms. It binds soil pollutants and it buffers the soil pH value. And it stores greenhouse gases. So losing soil organic carbon, soil organic matter, or soil organic carbon, soil organic matter is one of the key parameters in the soil health concept and of large relevance for sustainable soil management because of all these positive benefits. So here biochar can help. So biochar, organic waste produced by pyrolysis, high carbon content. So if we apply biochar to soil, we have an immediate increase in soil carbon storage, and that depends on the amount we apply, and this is less site-specific, and that is an advantage compared to other management strategies. We have a long-lasting effect because more than 90% is stable for more than 100 years because microbes are not really interested in decomposing this stuff. And we cannot have this benefits by a one-time application. That depends then on the amount what we apply, and it's not restricted to the surface. You can also apply it to the subsurface soil. And we have a variable organic feedstock, so we can use wood, corn stover, green cut, and so on. And here's just a little slide figure showing if you put manure, compost, and so on, that's all good for soil, soil organic carbon, soil structure, but you lose a certain amount by decomposition, so you're reaching a steady state. And that's not the case with biochar, because here the application rate or the input is bigger than the output or decomposition. That is because microbes are not really interested in decomposing it. We have further co-benefits, increase in soil pH, increase in soil water nutrient retention, lowering bulk density and nitrogen losses, improved soil structure, and we have an habitat for soil microorganisms. So, and how do we know that this works in the long term? So we have, so the entire biochar movement or recognition of biochar goes back to the discovery of so-called terra preta soils, that are soils formed and shaped by indigenous people a couple of 100 years ago in the Amazon, but also in tropical Africa. They mixed into their soils biochar or charred material beside excrements, bones, and so on. And we still see the soils 800,000–900,000 years later. That is a natural soil in the Amazon, a ferrosol, low carbon, low pH, really unfertile, and that's a terra preta. So we still see these terra preta soils in the Amazon and in other areas, so showing us that this is a couple of 100 years old and still present and formed and shaped by the addition of biochar. We also have sites in Europe, that is in an agricultural field in Belgium, 200 years of intensive agricultural usage. And here the black spots that were former kiln sites. So kilns were used to produce biochar for energy purposes. So that was all over in Europe. And 200 years later, if you sample this separately, you see a significant increase or significant higher carbon and nitrogen contents, but also calcium contents. So that is another example that is impact and influence and application of biochar is really a long-term process. So we have this natural kind of field experiments in the terra preta and here with the kiln sites, giving us in the direction and proving actually that biochar charcoal is a long-term solution. Then we have the nitrogen, the nitrate problem. So that is a map probability that groundwater source public water systems or private groundwater systems located here will violate the nitrate standard, which is, I think, 10 ppm. That is here where I live. So we have in Nebraska, we have areas with high and too high concentration of nitrate in the groundwater. And here are a couple of numbers for the US. So 11 million tons nitrogen per year fertilized, which is about $10 billion in costs. You're losing via nitrate leaching between 8 and 30%. So that's up to $3 billion lost in nitrogen fertilizer. So this nitrate goes in the groundwater, then it's causing public health problems, which can we amount to up to 6.5 billion per year. And we have more than 5 million people in the US with nitrate concentration above 5 ppm. So here we have an increased cancer risk. And this affects about 18% of population with self-supplied drinking water in Nebraska. So this nitrate leaching is another big issue in areas that are intensively used for agriculture. So, and if we look at biochar nitrate, so there was a pretty cool meta-analysis in 2019 from Biochar et al., and they showed large variation and opposite trends, but nitrate leaching was reduced on average by 13% in arable soil receiving biochar. There is a biochar time effect on nitrate leaching, so that means it takes a while until you see an effect on reduced nitrate leaching if you apply biochar to soil, and the underlying mechanisms are widely unknown. We see similar effects for phosphate, and this is linked to iron oxide. So that was one direction, or that's one direction our research is going. If you look at biochar and field application, you also need to look at the scale. So the scale is important. If you start at the nanoscale, we see changes in surface chemistry of biochar particles. There are interactions and redox processes at the biochar soil and biochar water interface. And this changes at the nanoscale that is influencing the changes in retention of water and nutrients throughout the soil profile. That's the soil scale. And that in turn is affecting the net nitrogen use in systems, crop uptake and yield, and in carbon sequestration. And if you go all the way back, you go on a field scale, and of course your irrigation management, your fertilizer type, and your management decisions are affecting soil biogeochemical environment for the biochar. So that is kind of a scale cycle. And if you really want to understand the benefits of biochar and predict the benefits of biochar at the field scale, so you need to, it would be a good approach to look at different scales and tie this to the different management forms we have in agricultural systems. So that is rather complicated, right? This is a plant, biochar, soil management interactions across scales. So our objectives, we are looking agronomic scale, we are looking into soil organic carbon storage and crop yields, we are looking at the soil scales in nutrient and water retention, and at the nanoscale, we are looking into biochar surface properties and with potential nutrient retention mechanisms. So that is our approach to analyze across scales. So our first two field experiments that were established in 2022, one is that are research farms in Nebraska and Lincoln, or close to Lincoln. One is in Mead, Nebraska, that is rain-fed. The other one is Clay Center, that is irrigated. And here you see some pictures, so it's rather small scale, how we applied the biochar. So that was with a forklifter and then distributed and then disked in about 10 to 15 centimeters in the soil. And so it's a silty clay loam soil, both of them similar texture. One is rain-fed in NREC, that's Mead. That is the rain-fed site, irrigated site is Skull at Clay Center. Both were corn-soybean rotation and conventionally managed. We had the winter cover crop and rye, and here we applied 70 tons per hectare or 28 tons per acre. So the idea was here to increase the carbon content by at least 50%. This is what we have lost over the last 300 years. So that was our study design. We used an existed field experiment that looked into cover crop effects, and then we split the plots and it could analyze the effect of cover crops with and without biochar and without biochar. So we had no biochar plus biochar in the cover crop system, and a control system where we had no cover crops, also with no biochar and biochar. So we could look into synergistic effects between cover crops and biochar. So then the feedstock. So we did a meta-analysis that was published by Li Dong Li, a former postdoc in my group. So here we looked into biochar from different feedstocks, corn, stover, switchgrass, and wood, and looked into low temperature and high temperature. And what we did find out was that the biochar carbon highest percentage is wood, high temperature, and that especially the specific surface area is much higher than in the other combinations. So for us, we wanted then to go for wood biochar produced at high temperature because we have a high carbon content and a high specific surface area, which is good for nutrient and water retention. So we have another problem in the Great Plains and especially here in Nebraska that is called woodland expansion, and that is the woodland expansion by eastern red cedar. So this is 1990, that is 2020. So you see how big the spread of the red cedar and grassland or rangeland encroachment is, which is a major threat to the beef production in the Great Plains, which is about 60 billion annually. Right? So if you look over here, the biggest conversion from grassland or rangeland is land use, so converting it to agricultural land, but the second big one is a tree cover expansion. So red cedar is for us a long-term solution and a long-term feedstock because it's basically killing two birds with one stone. So we get rid of the red cedar and we can produce biochar from it. So and at the same time, then we're protecting the grassland and the rangeland. So we decided to go in this experiments for high temperature biochar from red cedar trees. Okay, that were our methods. So soil sampling, first year down to 90 centimeters, up to 10, 10, 30, 30 to 60, 60 to 90. We are looking into nutrients, cation exchange capacity, bulk density, pH, but also, of course, carbon and nitrogen storage, nitrate and ammonia. And then we are looking into biochar particles, how they change. And here we use X-ray photoelectron spectroscopy, which is surface sensitive and gives you information about the chemical species at the surface. We also do soil carbon fractionation and delta 13C analysis to see differences in microbial processing, for example. And we also do FAME and DNA analysis for microbial community structure. Okay, that is the result 6 months post-application in SCAL and NREC. So 2022 in spring we applied. First measurements were done 2022 in fall after corn harvest. That is the NREC side, that is the SCAL side. NREC is rainfed, SCAL is irrigated. Here's a cover crop system, here's a no cover crop system. And the orange one is biochar, the green one is no biochar, and here you see the depth, 0 to 30, 30 to 60, 60 to 90. Not really surprising, after applying 70 tons per hectare biochar, we see a significant increase of soil organic carbon stocks up to 200% in the topsoil. If you look a little bit closer into this data, so we applied about 58 tons per hectare carbon, right? So that is, here you need to take into consideration the carbon content of your biochar and your moisture content. So we applied 70 tons per hectare biochar, which applied 58 tons per hectare organic carbon. What we could recover, what we see as an increase 6 months later was here 61, so that's good. 51 is good as well, 41 is okay, but here we only see an increase of plus 21. So that means about 35 to 40 tons per hectare carbon we lost. Our first, and that is critical, right? So if we apply biochar and if the community is applying biochar to their soils for the co-benefits for production, but also for carbon storage, we need to make sure that we do find this biochar again, right? So that the credits are getting in. Here in this case, we lost a lot of biochar. So the biochar was kind of powder form. So we lost a lot via wind erosion. So we developed a little protocol to avoid this or to reduce this. But that is critical, that the biochar that's being applied, that needs to be found again, right? Because biochar still is very, very expensive. So it's a long-term investment, maybe a once in a lifetime investment, but we need to make sure as a research community that we develop protocols that we actually do see the increase by close to 100%. So minimizing the losses at all costs. What we also are interested in not only bulk soil organic matter, but in soil organic matter fractions. Here, two examples, particulate organic matter and mineral associated organic matter. So the particulate organic matter, that is turning over more or less a little bit more quickly. So it's 1 to 50 years. So it's more important for soil formation, microbial substrate. And then we have some mineral associated organic matter that can stay in soil for thousands of years, right? So we have different fractions with different functions. We also see dissolved organic matter or water extractable organic matter, which is highly important as a transport medium, but also as a substrate for microorganisms. And it's kind of an, going back and forth between POM and soil solution and mineral associated organic matter. So it is kind of a transformative fraction over here. These are data from experiments we did in Germany. So here, that's a very sandy soil. We applied 0, 2, and 40 tons per hectare biochar. And here you see the free particulate organic matter, so quick turnover, occluded particulate organic matter, so organic particles occluded in aggregate, so very important for soil structure. And here are microaggregate and mineral associated organic matter. So in the stabilization protection, that's increasing if you go down. So just focus on this one. Here's the 0, 2, and 40. So you see a large increase of free particulate organic matter by biochar, right? So biochar particles, but also occluded particulate organic matter shows biochar. And here, the mineral-associated organic matter, right? So smaller than 20 microns. So a part of this biochar shows also up in the mineral-associated organic matter, which is an additional stabilization mechanism. So everything what shows up here is locked up for a long time. So here we have the low decomposition of biochar plus an additional stabilization in small aggregates or interaction with minerals. So that is pretty important also to see which different soil organic matter fractions are affected by biochar application. In our study sites, we are looking into particulate organic matter, occluded organic matter, water extractable organic matter, and mineral associated organic matter. All fractions can be affected by biochar, but also by cover crops. That are first data, water extractable organic matter. So that is not affected by cover crops or by biochar or by study sites. So that is pretty similar over here. Right now we are looking, or we are analyzing the other fractions, so POM, occluded POM, and mineral associated organic matter. That is the soil nitrate retention. So again, NREC, rainfed, SCAL, irrigated, cover crop, no cover crop, biochar, no biochar, topsoil, 30 to 60, 60 to 90. So in here, 6 months after biochar application, we have seen across the 2 study sites and across the management systems an increase by at least 11 kilogram per hectare in soil nitrate retention here in this field conditions. So, and this is under field experimental condition, conventional management. After 6 months, we see this positive effect on soil nitrate retention. And this is matching data from a greenhouse experiment we did a couple of years ago, published in 2023 by Jen Cooper. It's also a former postdoc in my group. Here we had a greenhouse experiment, and we looked into a sandy loam and a silt loam, fertilized with slurry, dairy or swine slurry. And we had, that is at harvest, right? So when we harvest the corn crops, and that is final, so 40 days later. And you see across the systems, across the two soil types and across the different fertilization regimes, in the final stage, biochar, significantly higher nitrate retention than non-biochar soil. So application rate was here 40 tons per hectare, also high temperature wood biochar. And so that is basically, so we did see this higher soil nitrate retention in greenhouse experiments and in our follow-up long-term field experiments. This is the soil moisture, so 2022 after corn, so no effect. 2023 after soybean, we see an increase in soil moisture, especially here in the rainfed site at Scar. So that takes a while, but there is a positive trend, at least for 2023, that we have a higher moisture content in the biochar amended topsoil. So that is for the 0 to 10 centimeters. Then there's our first data from our FAME analysis. So that is microbial community response. So that's in NREC. That are the microbial or the microorganism groups. We looked into bacteria, microeukaryotes, AMF, and apophytic fungi. In general, so that is a little bit different. So it's no cover crop and cover crop. Here you have no biochar, no biochar, the different soil depths. In general, the community structure is not affected. What we did see is that the cover crops show an increase in saprophytic fungi, but no specific effect of biochar. But the biochar amendments increase the overall microbial biomass. So there was a positive effect on microbial biomass in the soils, probably by adding habitat, but also nutrients, maybe leveling the pH value. So there could be a couple of co-effects. The same data for skull. I have. Okay, thanks. I had a background noise. So same analysis for skull for the rainfed side. No differences in the FAME groups, but again, in this case, a higher microbial biomass in the biochar amended soil under cover crops, but not under no cover crops. But in 3 out of 4 cases, we see under field conditions, field experimental conditions, an increase in microbial biomass. So then we looked into the changes of the biochar particles in soil, and for this we used XPS. So that's a fingerprinting technique for identification of elemental composition and chemical and electronic state. It's a quantitative determination of composition, but it's limited, it's good. That's limited to the uppermost 10 nanometers of the surface. So that's really surface sensitive. So we use the biochar particles, the pristine biochar particles before applying to the soil. And then we also hand-picked soil samples and cleaned using sonication for 2 minutes. And then we looked into the pristine biochar particles and biochar particles after 6 months in the soil and looked how the surface composition is changing. And so that are data for iron. So that's a pristine biochar. So we don't see any iron on the surface. It's a pristine biochar. 6 months after application, we see Fe2 plus and Fe3 plus. So in our explanation for this is that over time, we have bi-redox conditions, a reduction from Fe3 to Fe2. Fe2 is soluble, goes to the surface, getting oxidized to Fe3 plus, developing iron oxides on the surface of biochar, which we call a self-functionalization by iron oxides of the biochar surface. And that takes a while and is probably promoted by the more regular fluctuation in moisture in the irrigated. So we had 50% of rain-fed biochar particles showed this cluster, and 95% of irrigated biochar contained surface iron. So here in the irrigation, we have more constant fluctuation in drying and moisture, maybe accelerating the the dissolution, reductive dissolution, and the movement of Fe2 plus, and then transforming to Fe3 plus. At the same time, again, that's nitrogen here, pristine biochar. That is the irrigated site at Skal. We see a development of nitrate. And there is a paper out there, Akalas et al., I hope I remember the name correctly, showing that nitrate can bound to iron oxides via outer-sphere and inner-sphere complexes. So they are forming stable complexes under neutral conditions. So that was shown in a paper. And our explanation here is that after formation of iron oxides on the surface of the biochar, that is enhancing the nitrate retention, because we didn't see this before. Right? The pristine biochar also does not show any nitrate. And then 6 months after, we see the nitrate. And so less than 5% of iron functionized particles at irrigated site contain nitrate. But if you see nitrate, there's always, so you don't see nitrate without iron. Sometimes you see iron without nitrate, but you don't see nitrate without iron. And the nitrate feature, that's a small peak, it's a nanoscale, but if you apply 20 tons per hectare and you have 200 square meter surface per gram biochar, you have 400 centimeters square of biochar in 1 centimeter cubic. So that can amount to a pretty high retention capacity. That is, again, data from our greenhouse experiments that were kind of done before we did the field experiments, but we have seen here, or we did see here the same patterns, or maybe other way around. In the field experiments, we see the same pattern as in our greenhouse experiments. That is an example for the sandy loam dairy slurry treatment. Here you have pristine, harvest, and final. Again, no iron at the pristine. And then during the greenhouse experiment, we see Fe2 plus, Fe3 plus, and we see nitrate retention. So what we have seen in our greenhouse experiment, we can confirm under field experimental conditions in our field experiment. Okay, some take-home messages and outlook from this experiment. That was a paper in 2024 by Acharia et al. Biochar impacts on soil water dynamics, known unknowns, and research directions. So they said it is critical to explore large-scale applications and conduct long-term field studies across different management, climate, and soil types to fully understand how biochar impacts soil water dynamics. We agree, but we would like to expand this to soil, carbon, nitrate, and microorganisms. The temporal evolution of biochar properties during aging necessitates long-term field studies to elucidate how biochar properties, including surface functionality, changed after a single biochar application, and how this transformation influences soil water dynamics. So this is exactly what we are doing. So we agree, and this is key to understanding the improved nitrate retention. Our next step was that we are looking into the reduction of biochar application rate by combining with an organic fertilizer. And then we established in Lincoln 60 acres field experiment. That's, to the best of my knowledge, one of the biggest field trials in the US. Here we combined biochar with biosolids. So we have a biochar treatment, 20 tons per hectare, biosolids, biochar plus biosolids and control. 16 acres, cover crops, no-till management. So that was the, so we applied more than 120 tons of biochar. That was a loading, that was a manual spreader, and that was a drone footage where we applied the biochar. So you can nicely see where the biochar was applied, and then it received biosolids or no biosolids. That is located over here. So that is our third field experiment. That are the first data. So we don't see, that was after corn harvest. It was not fertilized at all. So the biosolids plots didn't receive any additional fertilizer. The other plots did, but we don't see any differences in yield. So that was good. For the biosolids, we saved 1 season complete nitrogen fertilizer and with no differences in yield. That is nitrate and phosphate retention. So especially the biosolid and biochar combination stands out over here. So that is basically supporting or showing that if you go down with a biochar and you combine it with an organic fertilizer, you can have positive synergistic effects. But here we are investing more money and analysis to look into more details explaining this. And this was the soil moisture content, June and July in 2 years. So also positive effects on the applications on soil moisture content. No consistent trends, but over here you see all 3 biosolids, biochar, biochar plus biosolids, positive effects on soil moisture content. But again, a little bit of a standout, the biosolids plus biochar seems to be effective in increased retention of moisture. Okay, that are our outreach programs. So we are present at field days, UNL research farms, discovery days, UNL campus coverage and farmer magazines. We have a citizen science project for urban soil health. That's a map with citizens in Nebraska. So as I also apply biochar. We had the Backyard Farmer YouTube channel podcast, then an INR podcast with a market journal, and we do also have a biochar project at a high school in Lincoln. And we organized last year together with Nash Leaf, Kim Slesak, and Frank Ularic, the first Great Plains Biochar Conference with over 70 people gathering in Lincoln. With this, I'm done for today. Thanks for coming and for your attention, and I'm happy to answer any questions.
Michael, thank you very much. That was a great presentation. Very glad that you came here today. We do have some questions in the boxes here, and I'm just going to scroll down here to find them first.
Yeah, so thank you so much, Dr. Kaiser. I gathered the questions that everyone put in the chat while you were presenting. So the newest message in the chat has 7 questions. And then everyone else feel free to add more in the chat, but those from the presentation are here so we don't have to scroll.
Fantastic. All right, starting with thank you, Katie, for putting that together. Number 1, fewer volatiles and greater pore size with greater temperature in biochar as well. Is that correct? So at higher temps, do you see fewer volatiles and greater pore size?
So the volatiles go down and the specific surface area goes up. Pore size is more difficult because you would need to look into pore size distribution. That's tougher to measure. So average pore size distribution I cannot say, but specific surface area goes up. That is usually a function of micropores, and usually the volatiles go down with higher temperature.
Fantastic, thank you. Next question, is the red cedar an invasive species?
It's an aggressive species. So invasive species would mean it's not natural to Nebraska, but it is an aggressive tree species. It is often described as an invasive tree species, but my colleague Kim from Nebraska Forest Service told me it is an aggressive tree species but it is encroaching. That is a main tree that's a problem in Nebraska and the Great Plains.
Fantastic. And what is the difference in surface area between soft and hardwoods?
I cannot tell you from the top of my head. So we in our meta study we did not differentiate between soft and hardwoods because it was hard to define, at least for us. So I don't know. I would need to look in the literature. There might be some reviews or meta studies about that. So we didn't look into that. So I don't know.
Fantastic. All right, getting back here. We were on, okay. Due to its longevity, doesn't the biochar just go and be helpful somewhere else? Consider it a community improvement. Think about the migration of biochar from your site.
So the question is if we are losing biochar from our soils.
Yes.
So that's a good question. The biggest, so after application, what we do is we disc it in. So we get it into the soil. So it needs to be mixed with the soil, that you have the biogeochemical reaction, including iron oxides, microorganisms, and so on. What much more important is if you would apply it in the surface, it's much more prone to wind and water erosion. So once you applied it to your topsoil, you need to make sure that you minimize your erosion. But this you should do in any case, right? So erosion should be avoided at any means in any system. So we are then, after biochar application, we go back to no-till or reduced till, and we do have our winter cover crops. So we try to reduce the erosion as much as possible. A second way of losing biochar is decomposition by microorganisms. You cannot do anything against this, right? So that's their job. And leaching could be over time if biochar is it's not inert, right? So particle size goes down, and weathering products, so you might see that particles migrate down the soil profile. But I wouldn't expect leaching losses as too high, because water extractable organic matter on biochar is pretty low. It would be more like a translocation. Nevertheless, we see in long-term field experiments that you might lose biochar, right? So, and most obvious is erosion, but also in part decomposition. So it's not entirely inert, right? So once the microbes are settling at the surface, they decompose, and usually the low temperature biochar is better decomposable than high temperature biochar. So it's a complicated question that, you know, there is not a single true answer on that.
Fantastic. And the fifth question. So do we know which phyla of organisms are taking up the additional nutrients? Is it limited to a specific phyla and could be more beneficial to them versus others? And I'm thinking about where exactly this could be applied. It is specific to plant, and if it's a specific to certain plant communities.
There are for sure data on that. So we didn't see differences in community structure based on fame. We did DNA analysis that is being processed right now. Biochar itself is low in nutrients, so there are almost no available nutrients. It becomes interesting over time if the biochar is charged. So, and we do not co-compose our biochar. We just put it in the soil. We want to make it as you know, as easy as possible. That is more a question like, you know, after a couple of months, if biochar starts to take up nitrate, phosphate, and other nutrients, how this can be how this affects the species composition. So based on our data, we didn't see any differences. Once we have the DNA data, we might be able to answer the question for our system. But there are for sure data outside looking into this into more detail. But on the top of my head, I'm not aware of any phyla.
Okay. Sixth question. Does the biochar influence the proportion of fungal microbes compared to bacterial microbes?
Based on the FAME data, after 6 months, we didn't see an effect. That might change after 12 or 18 months. So we are doing FAME analysis continuously now, so at least every year. So maybe the data for after 12 and 18 months look different. based right now on the FAME data we have, we did not see an effect for our systems.
Fantastic. And the last question here, we have some hands up. Was the biochar charged with an inoculation in any way?
No.
Okay. That one's pretty straightforward. And looks like we have Marshall has his hand up. So what's your question, Marshall?
Can you hear me?
Yes, we can.
Okay, so several things. I think you just answered one of them. You're taking biochar right out of the machine and putting it into the ground.
Yeah.
That's one thing I understood. The other thing that's not clear to me, I think your time frame is 6 months maximum, and you said that you're going to look at it after a year, maybe 2 years or 3 years. Is that my understanding?
Yeah, so we applied the biochar, and now the field experiments are running, and we are sampling 1 or 2 times a year and doing as much analytics as we can. So data I presented today were 6 months after application, but the next data we are looking into are then 12, 18 months and so on. So depending on if we sample in spring or fall.
So our experience, based both scientifically and also practically in Colombia for coffee crops and things like that, and cacao, is when we apply biochar, we apply it as a soil amendment that is formulated with other materials based on the soil biome. Doesn't sound like you're doing that. Number two, we measure over time, and we have found that if you put soil biochar as is out of the machine, depending on what the feedstock is, you're going to get degradation in the soil biome. It's going to leach out all the materials that you're getting good results on now in 6 months, a year, but after 3 to 4 years, maybe even 5 years, you start to see degradation. And we've been doing this for over 10 years. And so I would encourage you to continue to measure this, but also consider formulating your biochar based upon the soil biome. It's kind of like a drug. You don't give a gram-negative drug to a gram-positive, infection. So, or vice versa. So that would be my recommendation to you.
Thanks. That's a good point. But the thing is why we will follow your recommendation and measure this on a regular basis as long as possible. The thing is, if you go for large scale application, which is kind of one of our main goals, our idea was to make it as easy as possible for the farming community. And if you apply 120 tons or 270 tons of biochar and you want to pre-inoculate this, that's almost not doable for large scale application.
Actually, it is doable, but we could talk about that offline if you want.
For acre wise, that you, yeah. Okay, good.
Fantastic. Thank you, Marshall, for your insights there. And we have another question from David. What did you see in regards to soil pH adjustment in the soil with and without biochar?
I would need to look back in the data, but either we did see a slight increase or no effect.
Okay. And Gabriella has a question. Have you looked at different application methods to reduce loss over time?
No, we did not, but we with our large field experiments, the 16 acres, we used biochar with a much higher moisture content and with a larger particle size. So we would recommend to have at least 80% between 1 and 4 millimeters. And that should reduce the wind erosion losses significantly. So what we do now is, so we look at the weather, of course, and we use a different physical characteristics for the biochar that should help in reducing the wind erosion.
Great points. And we have Nick asking, have you utilized biochars produced with iron filing inclusions during pyrolysis, or have you only explored accumulation of iron oxide in situ?
The in situ. So we do not pre-treat our biochar because the production units where we did get the biochar from, that's not, it's more like would be a laboratory experiment, but again, our motivation is to keep it as simple as possible and use actually the soil as our biogeoreactor. So we did not.
Oh, great points. And how long do you plan to continue the tracking of the effects on the fields? One of the missing data points that concerns farmers I've talked to is the history versus fad of biochar utilization.
As long as possible. As long as we get funding, we continue to sample and continue to measure.
Okay, this may be a repetition, but this is from Sarah. I was under the impression that adding biochar straight to the soil would immobilize available soil nutrients for plant uptake. Do you have any type of inoculation before applying the biochar? And if applying biochar without inoculation, is it essential to fertilize throughout the growing season?
So again, we didn't inoculate. Our biochar plots receive normal fertilization rates. So we didn't see differences on yields. But for example, our big field experiment where we applied biochar plus biosolids, we didn't fertilize at all with mineral fertilizer, and we didn't see any differences in yields. So the biosolid was not pre-mixed. It was first biochar, then biosolids. That might have helped with the inoculation. So we do not have experiments where we applied biochar without any additional fertilizer, and I wouldn't recommend this. Our aim is that we apply biochar and make recommendation that the farming community might reduce the nitrogen fertilization because we have a better retention of nitrate. But no on inoculation, yes on fertilization, and no differences in crop yields in any of our treatments.
And we have Jackie asking, would it be good ag practice to apply biochar after harvest to take up excess nutrients to avoid leaching, especially if not using cover crops?
If the mechanism that we are analyzing with the oxide formation on the biochar surface is responsible for nitrate retention, you probably need to wait a year until you see an effect. But there are other mechanisms postulated that biochar retains nitrate by physical entrapment. Means the water goes in the micropores, and the micropores keep the water from leaching out, and thereby keeping the nitrate. So yes and no. The iron oxides that might take a while to develop, the micropore retention that might work immediately. If a farmer would ask me, I would say apply it in spring. Then you might have probably chances for both mechanisms, so micropore retention and chemosorption at the surface.
Fantastic. And we have Gudmundur. Was the biochar carbon characterized as how much is in labile versus recalcitrant form?
The more labile forms is usually the volatiles. So I would need to look into if they analyze the volatiles, but it was very low. So the biochar produced at 750, 800 degrees Celsius contains almost no stuff that would be immediately available for microorganisms. And everything else, the microbes are not really interested in. Only if they colonize the surface a little bit, but I don't have the data in mind if they looked into the volatiles. But again, as high as the temperature, as lower are the volatiles.
That's a great question, Goodmunder. And we have, and I apologize if I don't pronounce, I want to make sure you get your name right. Ikra is one of our longest travelers here today, but he's asking, hello, everyone. How can we confidently claim that biochar is cost effective, especially when it needs to be applied in tons per hectare, which could pose significant cost challenges in countries like Pakistan? And how can we convince the farming community of its long-term benefits?
That is a good question. So one of the main obstacles for biochar is still the cost. So we are, if you buy it, an industrial scale production in the US, you're paying between $300 and $2,000 per ton, which is for many farmers still cost prohibitive. If you count in case let's assume you reduce your nitrogen fertilization by 50%, that would add to the benefits. If you reduce your irrigation by 20%, that would add to the benefits. If you so in the US, I know that's it's not completely tackling your question. You also have NRCS incentives, so you get money in. If you get money in via carbon credit programs, so it might help. In rural areas, there are technologies, low-tech technologies, and I know there are also mobile units developed. That would be more like what kind of waste do you have on your farm, on your field? You could produce biochar and put it into your soil. Convincing a farmer is always, increasing resource efficiency. So paying less, getting more. And there are a lot of literature outside saying, okay, we increase our organic carbon. I mean, that was the first aim, right? To produce biochar, apply biochar to increase carbon sequestration, and all co-benefits are developed afterwards. So that is the costs are still a problem, but there is development, so it's reducing. For more rural areas, low-tech mobile units using local waste resources. And then, so based on my knowledge, farmers talk to each other, right? So if it works in one field, so less fertilizer, less water, same yields, then maybe other farmers are adopting this. But that is an entire field that's we are working on. So it's a good question. It's a really, really good point.
No, I agree. And again, I apologize if I get the name incorrect. I'm going to try my best, but Sycot, what is the EC content after application of biochar? Is there any difference?
We didn't measure, but EC of the raw biochar was low. Also available sodium. So I looked specifically into that because of the biochar. So we didn't measure EC, but I wouldn't expect big differences.
Well, fantastic. I think we're at the end here for the questions. And John Webster, are you still on? Okay. If not, make sure you take a look. He posted some good notes on subscribing to the USBI newsletter and information about the conference that'll be coming up in Minneapolis. There's a number of other things here as well. Please take a look. Katie, any last message on availability for, see, you're answered a lot, but on the availability of recordings and getting on the Char Talk invitation list? That's our main means of going back and forth with folks.
Yes, so if you are interested in coming to this monthly webinar, again, it occurs every third Thursday at noon
Friday.
to 1 p.m. or Friday. Thank you, Jim. I have Friday brain. Every third Friday from noon to 1 p.m. Central Time. and we communicate that through the Char Talks email group. You can drop your email in the chat here or email me. I'm putting my email in the chat again. And we will send out a recap email as we do for all Char Talks that will be coming out at the end of next week. Jim mentioned John Webster's posts about the USBI. I also pinned a chat from John where he links a general biochar communications masterclass. It's on YouTube and a podcast. That's a great resource. And thank you, Jim. Thank you, Dr. Kaiser.
Yes, thank you very much, Dr. Kaiser. It is greatly appreciated for your time. This was very helpful. We had a lot of interest and a lot of questions, so I can't extend my gratitude enough for your thing. And Sebastian, thank you very much for recommending Michael to us for this Char Talk. It turned out very, very well and very much appreciated. So with that said, every third Friday, get on here. If you have, please let us know if you want to be included on the repeat Char Talk extended list. A lot of you came through the LinkedIn app. It'd be great to have you on, have it on a standing calendar. And we get a lot of people as the quality of Dr. Kaiser will be doing well. Thank you very much, sir. You have our gratitude from MNBI in sharing this knowledge. And I hope you have a great start to the year. And I look forward to seeing you again on Char Talk. If you have a topic you want to talk about, please let us know. We'd be glad to have you on Char Talk. So thank you very much, and we'll have a good day.
Thank you, everyone.
Thank you.